I'm going to talk to you about motor neurone disease, a problem that's consumed me for the last 25 years and continues to do so. And it's something that's probably, if I gave this talk 20 years ago, many people wouldn't really know much about or have heard about, but it's become much more prominent. Now, if you were in Chris Whitty's talk, you will have got the message that we're living longer. And that's an amazing thing. But what it does mean is that there's an epidemic, possibly even a pandemic, since it's certainly global, called age-related diseases of the nervous system. Alzheimer's disease, Parkinson's disease, and motor neurone disease, much less common, but they share the characteristic that they become more common as people get older. So why is that? We need to understand the aging nervous system in the context of these diseases. So here's a human story. I mean, these are statistics. This is a human story you may be familiar with. Doddy Weir was an international Scottish rugby player, a British Lion. He retired and, went and back to his farm in southern Scotland. And at the age of 46, started developing weakness in an arm, in a hand. That progressed. It progressed to his other upper limb. Then his speech became slurred. He lost the ability to walk. And he died after 6 years. And the image of him a few weeks before he died is deeply tragic. This is what the disease does. It takes somebody with an intact nervous system in the prime of life, and it dissolves it into one of no function. Here are some other characters from history that died of motor neurone disease. Charlie Mingus, jazz bassist. Lou Gehrig there is and if you go to the United States of America, everybody knows who Lou Gehrig was, even though this was almost 100 years ago that he was at the height of his fame. They call it Lou Gehrig's disease. Dmitry Shostakovich, Ronnie Corbett, Roberta Flack, David Niven, and Stephen Hawking. So the terrible fact is that we have done nothing over the last 30 years to shift the overall mortality. So 50% of people with motor neurone disease have died by 22 months from diagnosis or 30 months from their first symptom. So it's a highly malignant disease. 10% of people die in the first year. So Stephen Hawking was not the best poster child for the disease because he had it for 50 years, and it is not like that. It's also a disease that affects cognition in probably half of our patients, and about 5%, that's a dominant problem. 1 in 400 people will get motor neurone disease, which is probably a bit commoner than people imagine, even if there are only 1,300 new cases a year and about 5,000 people living with the disease in the UK. Now, if you take, so that previous graph here looks like it all obeys a certain sort of linearity. This is the mess, though. If you actually take individual patient trajectories and you look at those, you see this, what's called a spaghetti plot, meaning that there's nothing very easy to discern in there that ties all these people together. So probably that means that the underlying biology of the disease is immensely complex, and people live long periods of time or short periods of time for quite complex reasons. And that's pretty much what we've now understood and I'll tell you about. But it makes it difficult. So, what is going on here is that there is a system in our brain and spinal cord for voluntary movement. And if I wish to make a movement with my finger, there's a lot going on. There are cells up at the top of the brain which descend as a tract called the corticospinal tract, which in humans has 1.1 million fibers. And even our closest relative chimpanzee is a primate, from whom we are separated by about 5 million years of evolution, only has 800,000 of these. And so what's that all about? Well, part of it is for dexterity. And we know that because these connections are direct connections. They don't go via any other routes. Whereas almost in all other species, the cells that determine movement go via different sort of way stations in the nervous system. We have direct connections, and that may impose a kind of vulnerability in the system that makes it really vulnerable to aging and to loss of energy during aging. And, I need to, as a researcher, how do I approach this problem? Well, I'm a cellular biologist as well as a clinical neurologist. So I have a laboratory where we take cells from patients and we study them. But of course, I know this is happening in a broader system, but I have to start with a cell. This is a wonderful picture of what's called a pyramidal cell by Cajal, who got the Nobel Prize for developing the methods for how we can visualize these nerves. Yes. And you can see that this is a cell body here with tremendous ramifications everywhere, lots of connections. And in a spinal cord, if you inject dye into the cell, it is a tiny thing, but it has a really large kind of network of connections. So these cells are very different from blood cells, which are like a little sort of, egg with a nucleus in the middle. It's a white blood cell, and it's very small. It's 7 microns or 7 millionths of a meter long, a nerve cell. This nerve cell that I'm using to wave my foot around starts in my spinal cord, ends up in my foot. That's a meter. And yet the cell body is only 30 millionths of a meter in diameter. So if I were the cell body and my end, it would be 30 kilometers away. Mm-hmm. So this tiny process here has to travel, let's say, to a house in Banbury or somewhere, mm-hmm. and plug into the right house, into the right socket to make the element work, in which case it's muscle fibers. So how on earth that works in the first place is really something I still don't grasp. Mm-hmm. Now, this is an extraordinary paper. I mean, we can say the brain is complex, and we can wave our arms around and say, this is data that tells you just how complex the brain is. So somebody took 1 cubic millimeter of human brain, which they had harvested during a neurosurgical procedure, and they used the very latest imaging techniques to say, well, ask some very basic questions. How many nerve cells are in there? Are they all the same or are they different? How many supporting cells, which we call glia, how many of those are there? And how many connections are there? So in 1 cubic millimeter of brain, there are 150 million connections, synapses. So that, to reconstitute that, if they're falling apart, whether in Alzheimer's or motor neurone disease, is currently way beyond anything that we can achieve technically. That's an important message. Doesn't mean it's futile, but it's very, very distant, and it requires a technology to mature in a way that's currently not ready yet. So regenerative medicine, and my patients, are desperate. They all go off to all sorts of strange places and have people inject things into their brain because they're being told that, stem cells can make them better. This is not going to happen. So we need to work out who gets motor neurone disease, what's going on, and how we can really make an impact. So one way of conceptualizing these problems is by saying, well, if you look down a microscope, what do you see? And if you look down the microscope in motor neurone disease, you see very specific types of protein aggregates. They're called inclusions because they're not soluble, and they stain with something called TDP43. In contrast, in Alzheimer's disease, you see the staining with amyloid. So these diseases can be classified by the protein that you see under the microscope. Suggests they're different things. So one way into this is that in some patients who come to clinic, we take a very detailed family history from everybody. And in about 10% of cases, what we see is that there is a family history. So this is the person I'm seeing in clinic, and they will say, well, my mother died, we think, of motor neurone disease, and so did her brother. And their father, on his death certificate, it said the creeping paralysis is what they used to be called in the early 20th century. And this, you can see it's missing a generation here. So this person here should have developed motor neurone disease. Well, they might have died of something completely different because the age, the life expectancy has shifted upwards. But here in this family, there are people with different forms of motor neurone disease, all of whom have a mutation in the same gene, which is called SOD1. So we can start to model the disease by taking these families and taking these genes, looking at the mutations and seeing what's going on. And now, this is the SOD1 gene identified now a long time ago, 30 years ago. And now we have all of these different genes which cause motor neurone disease if they're messed around with by nature in families. It's immensely complicated because they all seem to do rather different sorts of things. These are just different cellular processes that relate to their function. So that's complicated. Even though you have that complexity at the genetic level, if you look down the microscope, 97% of the patients that we see in clinic have this TDP43 protein in the wrong place. So there it is in the nucleus of the cell, where it's involved in regulating the way that genes are expressed. And here it leaks out into what's called the cytoplasm, the area around the nucleus, and here it's aggregating. And so this is tempting to say, well, this is the disease when you look down the microscope. This is very, very striking. It has been likened to a dog barking at a bright moon. Is it the disease? Because actually that very long cell that I showed you earlier on, 99.8% of the cell volume is in the process, not in the cell body. So what's happening up here is likely to be very important, but it's absolutely not the full story. We need to understand the cell in its complete architecture, which is very, very challenging. Okay, so family history does occur in about 10% of the patients. 40% of those will have mutations in this gene here called C9orf72. It's simply a postcode, chromosome 9, not a very elegant name. And here are the other mutations. So these are the ones that are important because they're common, and TDP is important. This is the gene for TDP because if you mutate that, you get a disease which is exactly the same in the clinic to all the other patients. I can't distinguish it until I do the gene sequencing. So that means if I work on models which have TDP43 mutations, and I work on models with C9orf72 mutations, both which have the same pathology, I might get somewhere. So we have a clinic here in Oxford who set up 25 years ago. So we see about 10% of all the people in the UK who have motor neurone disease. They come from far and wide. And what we're trying to do here is to look after them as well as we can, but they are the partners in our research. And patients are highly motivated. They come from all over the place because they want to help. Even if they know this is not going to benefit them, they want to be partners in making this history, if you like. So we do this by trying to understand patients and all their complexities. So we use big data from UK Biobank, for example, where there are now 800 people who've developed motor neurone disease in the last 15 years or so. We use the fluids that we get from patients in the clinic, blood and spinal fluid, to do very, very deep proteomics, it's called, to look at what's the constituents of these things. Can we actually determine, is it one disease or lots of diseases? We use models in the laboratory, and my lab does that in the Kavli Institute here. And then we try and identify drugs that might improve the life and health of those cells that we're culturing. And then we go back to the patients to say, can we actually demonstrate this drug is engaging the disease in a patient? That's the basic principle. So one of the great advances really was, I was always jealous of hematologists, for example. You can take a bit of bone marrow out, you can see under the microscope immediately what's going wrong. And that's probably why a lot of hematological diseases have really had much greater progress than neurological diseases. But this happened, that we could take a skin cell, we can reprogram it into what's called an induced pluripotent stem cell, one that can give rise to any cell in your body, and we can tell it to become a nerve cell. And then, therefore, I can have nerve cells in my dish, in the laboratory, come essentially from the patients in my clinic. And that changed a lot. So we do that by giving a cocktail of different substances to mimic the way these cells develop in the human body. And we end up with this kind of morass of motor neurons in this case, which we know are electrically active, which is very important. So they're behaving like nerve cells. They can fire like nerve cells, and we can study what's going on. So Ruxandra Devinka is a postdoc in my lab, and was a student in the lab, has spent the last 10 years characterizing these in great detail. And she can show that if you take cells from patients with this mutation in C9orf72, which is the commonest mutation, there's a very small signal that they're unhappy, that they are showing cell death pathways and they are surviving less efficiently. But it's not massive. They're not dying in huge numbers. That's good. But something's wrong. What is going wrong? Well, she's gone on to use imaging to show that in these very long processes that I was describing to you earlier on, you can see a problem. So this is people with mutations in this gene TDP43. And we can label the mitochondria, little packets of energy-giving substance, which are going up and down this sort of train tracks in the nerve cell to supply, ultimately, the bit that matters, the bit that connects to muscle. And if you've got a healthy nerve cell, you can see these things speeding up and down at a great rate. In this cell, which is from a patient with a mutation, there's lots of pausing and there's not so much movement. So there's something fundamentally wrong with this system of delivering the right cargo to the right place. And it tells you that this immense length of the nerve cell is part of its vulnerability. And that's a very energy-dependent process. So we can show that in the presence of this mutation, the mitochondria are not working effectively to produce the energy that allows this transport to occur. So she's now going on to link the energy generation, the axonal transport, and what's happening right down at this, what's called the synapse, this connection between nerve and muscle, or between nerve and nerve in the central nervous system, to try and link those three things to find drugs that can actually work at each of those stages. Back to this immensely complex 1 cubic millimeter of brain, there's not just nerve cells in here. There are cells which are called microglia and astrocytes and oligodendrocytes. These are cells that provide support for the nerve cells. So even if we can convince this is a nerve cell disease, we have to understand what's happening in the milieu around it. So in fact, it turns out that nerve cells, if they become damaged, trigger activation of all these different cells, which might be helpful or it might be harmful. So Björn Varsson, who is in my laboratory, has pioneered growing together the nerve cells, motor neurons, with these cells microglia, which are thought to be very important in the inflammatory response to motor neurone disease. And what Björn showed recently is that if you culture together microglia with this mutation with normal nerve cells from somebody who didn't get motor neurone disease, you can see that the microglia are producing a toxic response, and it's due to this protein called MMP9, matrix metalloproteinase 9. And if you block it, you can actually improve the survival of the cells. So there is something that's coming from these other cells, these microglia, which is critical to the disease process. Lucy here is a clinical neurologist. Lucy has just finished her DPhil, and she is growing nerve cells into which she has put something called an optogenetic construct, which is a little bit of protein from another species, which if you shine light on the nerve cells, which is she's doing in this array here, and this is muscle, which is connecting to the nerve cells, and you can see that the muscle twitches. It will twitch anyway. Let's stop twitching. Hopefully you saw the muscle twitching in response to the light. So what we're doing there is trying to actually look at the whole system and say, well, looking at the nerve on its own in isolation isn't not going to give us the answer. What's actually happening at the connection? Can we work it out? And she showed that if you stimulate that nerve at high frequency, then you will get cell death, and that's mediated by various proteins and calcium signaling, and therefore it's an activity-dependent problem that's going on in these cells. There we are, more switching. Okay, so what we've basically been able to show using these kind of models is that if you actually stress these cells with something called oxidative stress, which is a very poor but a rather broad mimic of aging, as we get older, we become less able to cope with this kind of stress in cellular pathways. You will see impairment of this transport along these large axons, these projections in the nerve. You'll see various other phenomena that relate to that, and you'll see an energy generating problem. And ultimately, what that means is that the packages of molecules that are required to make the cell work at this critical point, the synapse, may not be processed properly, may not arrive there, may not be assembled properly, and may not survive in the context of stressful stimuli. So it tells you that there are a whole range of points of vulnerability, and therefore no one drug is ever going to make a difference. So it's my view. And it also tells you something very important about the disease and its complexity, because if you have one of these mutations, then you're born with it. In fact, you're conceived, and you grow and develop, and your whole nervous system develops with this mutation, apparently without a problem. You live for decades. You may be a very successful person physically. You may be an athlete. And yet, once the disease kicks in a way that can be seen clinically, you could be dead within a year. So that means that this is not a single hit disease. The gene mutation is not a sufficient condition to allow the disease to occur. You need these other hits to occur. These are age-related things that happen. What are these things? They're things that affect us as we get older, that may be occurring and causing local problems in our nervous system and transmits into a much more complex disease. That's difficult to model in the laboratory, but that's what we're trying to do. Okay, so this is a complex disease with a genetic background, development, changes, age-related changes in genes and cells as you get older, leading to this one pathology. When does this actually start? Does it start just before you might present to a neurologist? Or does it start years before? So UK Biobank is a collection of 500,000 people who gave blood samples and have been scanned and various things. I mean, I'm one of them. And over the last 15 years, I've gone and had imaging done. I've given various blood samples. We've gone to the biobank and said, people who go on to get motor neurone disease, are they actually the same 10 years before the disease starts? And you can see differences in their metabolism. That tells you that something's going on before the disease starts. But that's a good thing, because if it literally was something that came out of nowhere and then the patient comes to your clinic, it would be very difficult to see how you could solve the problem. This tells us we have the capacity to embark on a preventive program. How do we prevent the disease? That's very important. I'm going to skip this because I'm over time. But this study here, which we call ACORN, a C9orf72 national study, we're going to collect 400 or 500 people who are carrying the genetic mutation, who've had a first-degree relative with the disease, and we're going to study them longitudinally to see what are the very first signs of a disease using protein biomarkers, but also imaging to look at the way the networks in the brain are working. So we've already started that, and we've got 60 or 70 people already. We're getting about 10 a month, and this will be an incredibly important resource. Successful therapy is not going to be one blockbuster drug. This is a very incremental process, chipping away bit by bit. If you've got a gene mutation, actually, there is now for this SOD1 version, which is 2% of all the patients, there is a treatment which actually does have a significant impact in stabilizing the disease. But if you wait till someone comes to your clinic that's already got it, that isn't going to do anything except slow it down. So we need to work out these early signals that tell us someone, even with a genetic mutation, will get the disease in the next 5 to 10 years before we can give these very expensive and potentially toxic therapies. You can't give them to 10-year-olds. So even for these so-called precision medicine approaches, we need to understand that early phase before the patient will present to the doctor. But ultimately, most people will be treated by combinations of drugs, acting on all these different pathways that are active. And then prevention, above all, is probably, for all these diseases, Alzheimer's, Parkinson's, and MND, absolutely fundamental. So thank you very much for listening. We're very happy to answer any questions.
Hello, so I'm Hannah Smithson, and I'm fellow in experimental psychology. Now, experimental psychology is a very broad church. My specialist interest is in sensory science, sensory neuroscience, and I specialize in vision. So this is a very famous image, the London Underground map, but it's a disaster in terms of accessibility of design. So about 8 to 12% of men will have difficulty with this map. And I'm not claiming here that there are any gender differences in the ability to map read, but instead 8 to 12% of men will have some form of colour vision deficiency. So for those of you who don't have a colour vision deficiency, we can simulate what this map would look like to somebody who does have colour vision deficiency, and that would typically be colloquially described as a red-green colour blindness. So the dichromat simulation here is in this black box here. For the normal trichromat, the district line and the central line are very easily distinguishable colours. For the dichromat, they're very difficult to distinguish. Again, for the trichromat, the Bakerloo line is brown compared to the red and green of the central and district lines, but for the dichromat, they're all this sort of murky brown colour. So colour blindness doesn't mean that you can't see colours, it means that you can't distinguish between particular sets of colours. And in particular, you have difficulties with reds, greens, browns, yellows. Now, we know quite a lot about the underlying biology of this. So your vision in daylight depends on the cone photoreceptors at the back of your eye. They're known as cones because they have this kind of cone shape as part of the cell. And we have, in the normal trichromatic human retina, we have 3 types of cone cell. And they're different because of the wavelengths of light that they're most sensitive to. So we call them the S cones, short wavelength sensitive, M and L cones, middle and long wavelength sensitive cones. And to distinguish colour, you're reliant on a change in the relative activation of these cone types. That gives you information about the balance of different wavelengths in the stimulus that you're viewing. Now, we know quite a bit about the underlying genetics coding for the photopigments in these cells. So the gene for the S-cone pigment is on chromosome 7, and the gene for the L and M cone pigments is on the X chromosome, indicating why it's sex-linked, because men are XY individuals, whereas women are XX individuals. We have a backup copy of the gene. And these genes, the L cone genes and the M cone genes, are highly similar. It's thought that the difference between the L and M cones evolved fairly recently in our evolutionary past, only about 50 million years ago. And they remain genetically sort of 96% homologous. But it means that during the process of meiosis, they're quite likely to be misaligned or corrupted. And we know single nucleotide polymorphisms that knock out these genes. And so if you're a dichromat, for example, you're missing the M cone genes, or you have a corrupted version of the M cone genes, which means that you don't have functioning M cones, and you're missing this spectral sensitivity here. So that all you can do is compare broadly short wavelengths and long wavelength lights, warm and cold colours. So essentially on that simulation, that map looks like a series of blues and yellows, warm and cold colours. And that's all you're doing. If you're a dichromat, you've just got that balance. So I work on colour vision and colour vision deficiencies. We're interested in prevalence of colour vision deficiency and how that interacts with different ethnicities, different lifestyle choices can affect colour vision, not genetically, but through the pigments, additional pigments in your eye. So we're interested in individual variation in colour vision. But I've always had an interest in trying to get closer into the biology of vision and understand more about what's going on mechanistically when we see things. And so light comes into the eye through the cornea and pupil and is focused onto the back surface of the eye here, known as the retina. This is a zoomed-in version of the retina. The retina is actually a layered bit of tissue containing the photoreceptor cells that I talked about, cones and rods, but other processing neurons as well. And the axons of these neurons form the optic nerve, and information is transmitted to the brain. So if you go to the opticians, you might see an image like this. This is the optic nerve head, and this is the center of vision, where your vision is best, most acute. It's known as the fovea. So to try to see individual cellular processes in the living eye, we want to be able to look through the pupil, the cornea, through the lens, through all the other aqueous materials in the front part of the eye to get a good image of the retina. Now, you'd be familiar with looking at the bottom surface of a swimming pool through water, and it's very highly distorted. And so you don't get a good image of the bottom of the swimming pool if you're looking through all of this distortion. And astronomers have suffered a similar problem. If you have a ground-based telescope and you want to image a distant star, you've got to do that through the atmosphere. And so they developed a system called adaptive optics, where you measure these distortions and try to correct for them. So we start with a light source, we shine light into the eye, and we collect the light that's reflected from the eye. And we send that to an imaging device, to a detector. But as the light comes out of the eye, instead of being a perfect plane wavefront, it's now distorted, distorted by the optical aberrations of the eye. But if we use a mirror whose surface we can distort, we can put the opposite distortions onto the mirror and therefore correct them. And we do that using a process of also measuring these distortions and having a continuous feedback loop that measures the distortions and corrects them with the mirror. And when we do that, with a system like this, which was painstakingly built by one of my postdocs, it's one of two systems like this in the country. There are more in the States, but it's one of just two systems in the UK. And each element here has been painstakingly placed on the bench, aligned to micrometer precision, and we switch it on and we hope that it works. But it did. So it's essentially a fancy microscope, but it has all the technology available to correct for the optical distortions and get high-resolution images of the retina. Rather than in a microscope, you'd have the microscope and the tissue sample would be on a slide, mounted a bit of tissue on a slide. The tissue sample here is at the back of the retina, and it's in the living eye. So the very last lens of our system is the lens of the human participant. And so we ask our human participant to sit here, and the imaging is done through their eye. So in the living human eye, we can get images of individual cone photoreceptor cells. Each one of these blobs is a cone cell. We're looking en face, so we see the front projection of them. This is in central vision, quite near the center of the fovea, where the cones are very tightly packed. In peripheral vision, they're larger and more widely spaced. And if you go further out into the periphery, there are rod cells interspersed between the cones. So we can get very detailed information about the numbers of cones, degeneration in inherited eye disease, for example, that often changes the number of cones in the eye. This kind of imaging can give good information about degenerative eye conditions, such as those associated with diabetes, for example, or age-related macular degeneration. We can change our imaging geometry a little bit, and here we're collecting scattered light rather than the directly reflected light from the front of the cones. And I put this slide in because I wanted to show you the similarity between the images we can now get in vivo in the living human eye, non-invasively, where we see cones here and interspersed rods here, and the type of image you'd get in a histology slide. So these are the sorts of images I've shown you, but the actual data capture is a real-time video of the eye as it moves. And this is on a loop, but even when we ask our participants to fixate on a target so that we can image their eyes, they can't keep their eyes perfectly still. Their eyes are always moving about a little bit. And that's a bit of a nuisance in terms of imaging. To get these averaged images, we have to stabilize this image and then average them together. But it's also interesting in terms of neurological control of eye movements. So these eye movements cannot be suppressed, and we know that they're also under neurological control. So we can use this system to very, very accurately measure the movements that the eye makes. And we can do that also while we're stimulating individual cells, or even stimulating cells and moving that stimulation so that we keep hold of a single cone photoreceptor that we stimulate during an experiment. So it's difficult to imagine that when you look at something, you're failing to keep your eyes still. We have this subjective impression that we can control our eyes very well. So I wanted just to take you through visualizing your own fixational eye movements. So this is the target I'm going to ask you to look at. So try to keep your eyes steady on the black dot. And while I'm talking, just try to fixate on the center here. Now what I'm doing here, keep your eyes really still. What I'm doing here is trying to get on your retina an after image that's sort of colloquially burnt into your retina, so it's fixed on your retina. Now I'm going to remove the grid, and I ask you still to keep your eyes still on the black dot. And you should see an after image floating around, but you try to keep that after image still. You can't do it. It'll keep moving around, it'll keep drifting off. And that's the process of your fixational eye movements that you can't completely control. You can't completely suppress them. So if an image is presented on your retina, this might be the stimulus in the external world. And we know that presented on the retina, on top of these cone photoreceptors, it'll be blurred by the eye's optics. But it'll also be smudged and smeared over the retina through these motion distortion artifacts. And so these purple lines here are trajectories of motion that we measure. And so this would be the sort of distribution of photons on the retina that you get when looking at that original C stimulus. So one of the questions we have is whether these eye movements are a nuisance or a benefit. Now, I can tell you that if I set you up in the lab with a device where I'm measuring your eye movements and I have the ability to move a stimulus at the same time as your eyes so that I can stabilize that stimulus on the retina, after a second or two, you won't see a thing. It'll fade to nothing. And so that gives us a hint that these eye movements are actually really important in refreshing the image on the retina. Now, when I showed you the image of the cone photoreceptors before, the ones in the center of vision are tightly packed, but the ones further out in the periphery are larger. Now, I can't get you to stabilize your eyes perfectly. I don't have the equipment here to track your eyes and keep something stabilized on your retina. But I can ask you to fixate, and that's sufficient stabilization for the periphery of your vision because the photoreceptors are bigger there. You don't have so much acuity there, so a little movement doesn't matter so much in the periphery. So I've got a stimulus here, which is a series of blobs arranged around the periphery, and I'm going to ask you to look in the center. Okay, so just keep your eyes fixed in the center of this image, which I'll show in a minute. And what you'll discover, if you keep your eyes really, really still, is that you should begin to see a green blob moving around the circle. But keep your eyes as still as you can, and the pink blobs will disappear. Okay, and that's happening because you're holding your eyes sufficiently still that those photoreceptors and subsequent processing neurons are receiving the same stimulus all the time, so they adapt. They get used to that being there. And they never actually present anything green here. If you move your eyes away, you probably get lots of after images, but I can out for it. If you move your eyes away, all I'm doing is turning off one of the pink dots. Because you've adapted pink being there, if I take it away, what you see is white minus pink, which is green. So this is really an optimization problem. We have position uncertainty from the eye movements, but they may be critical in letting us escape from sensitivity loss due to adaptation. And so this is a collaborative project going on in my lab at the moment, and we're drawing researchers with a theoretical physics background, thinking about the mathematics of this optimization problem, computational modeling, and vision science. Now, other things we can do with this retinal imaging system. I've shown you this kind of image. This big black region here is actually the shadow of a blood vessel. So there are many blood vessels that run across the front surface of the retina, and we can change our focus to image into the blood vessel itself. So this is an image actually in mouse retina of flow of individual red blood cells through the finest capillaries of the retina. And this is a similar map now for human retina, showing flows at various points in this vascular network. And we're able to do this now by tracking and using particle velocimetry to track individual red blood cells as they move through these vessels in the living eye. And for those of you who have an interest in sort of brain imaging and interest in understanding the workings of the brain, this coupling between neural activity and changes in blood flow, and particularly in fMRI brain imaging, the blood oxygen demand. We can use the change in blood flow to give us information about neural activity and metabolic demand. So this is an example where blood flow is measured in this vessel here. And there's either a control condition, the beginning of this graph is stimulus onset, control condition, the participant upregulates blood because they know something's happening at this point, and you can present a flickering stimulus in this region too, and that's pretty much the same as the control condition. But if you present a stimulus in the upper half of the retina, which is fed by this vessel, you get a strong increase in blood velocity there over and above the general alerting increase. So we can begin to unpack neurovascular coupling in this way. We can stimulate the retina very precisely and measure the change in blood response. These are a couple of videos that I enjoyed. So the streaks here are red blood cells, but the little blobs are white blood cells moving along the inside of the vessel. And this second movie, it's a lovely movie with a white blood cell going out to deal with inflammation. It's an immune response to inflammation. And then the blood cells migrate back into the vessel and go off on their journey round the body. So this is a window into the immune response, again, actively visualized in the living eye. This is mouse retina, but it's technically possible to do this in human retina as well. So that brings me on to the final bit of my talk, which links quite nicely to Kevin's talk. So you might begin to see now that the retina is this really quite accessible piece of neural tissue. where we can get non-invasive optical imaging of very detailed information about underlying biological processes. And the retina has its own vasculature and its own neural circuits. So it potentially offers us a really nice window on brain health. And over the last few years, there's been a big rise in a field known as oculomics, which is using information from the eye to get information about other conditions. So, for example, these are some recent review articles: past, present, and future role of retinal imaging in neurodegenerative disease, biomarkers in neurology and psychiatry, insights into systemic disease, and that would often be vascular disease, through retinal imaging-based oculomics. One of the important things here is that potentially imaging in the eye gives us early warning signs about diseases that are about to develop. And it was sort of one of Kevin's points that it's very difficult to repair damage after it's happened. But if you can get early markers of damage, then you might be able to intervene either with lifestyle choices or drug choices to try to protect from further damage. Now, all of this research has been based on clinical grade imaging and large AI big data models to try to extract information that correlates with later disease. But the current clinical imaging isn't at this kind of resolution. And so one of the things we're working hard on with our collaborators is to try to get this sort of high-tech next generation retinal imaging more widely used, and probing that to get markers of disease, insights into disease. And so I'll end with sort of the state-of-the-art of retinal imaging with our collaborators in the States. So this is a AO OCT SLO instrument. So it uses interferometry as well as just reflected light. And we have a fly-through of a little volume of neural tissue here. And so the arrow tells you in depth how far you're going through the retina. These are the ganglion cell bodies. Come to some vasculature, bipolar cell bodies in a minute. More vasculature. And then when we get to here, these are the standard en-face images that I've shown you of the cone photoreceptors, which come there. But all of this can be obtained in the living human eye while somebody is sitting in our lab with no intervention other than asking them to sit still for quite a long time. So we know a lot about the early stages of vision, genetic, physiological properties of cones, and one of my interests has been linking that to visual performance. But this retinal imaging work means that we can stimulate an image at the single cone level, which means that we can unpick some of these neural circuits that support human vision. Imaging of retinal blood flow is a nice proxy for neural functioning. We also perhaps have a window on immune response, which may be implicated in various diseases. And generally, beyond using retinal imaging in vision science and ophthalmology, the retina potentially provides a useful window on brain health. Thank you.